Let me share with you some issues unveiling the secrecy of the upgraded model of healthcare services, where practically 90% of modern trends in bioengineering, chemical engineering, bioanalytics, and related sub-areas are being aiming. Personalized and precision medicine, drastically different from healthcare services all over the centuries. It's a new model of healthcare services, just as the unique entity, demonstrating an integration of fundamental clinical science, bioengineering, and translational art and IT armamentarium, whilst illustrating application of the different tools and 3 global levels: individual, population, and community. To achieve the implementation of PPM concept into the daily practice, it's necessary to create a new strategy based upon and driven by biomarkers. It's just the definition suggested and proposed by National Institute of Health 15 or 16 years ago. Biodesign-driven projecting, for reference. In Stanford University, this is the name of the training program, 2.5 years. In Cambridge University, 2 years, and at Heidelberg University, 1.5 years. Nothing more. Biomaterials is one of the crucial elements of the new trends. And finally, biotechnology as a whole. PPM as being now is known like the grand challenge to forecast, to predict, and to prevent, is rooted in a big and a new science generated by the achievements of systems and synthetic biology. Very briefly, just the illustration of the latter. And developmental engineering paradigm, including translational applications to connect basic research and daily clinical practice through a much complicated translational bridge. Translational bridge today and tomorrow, training course in Yale University, 2 years. Europeans do not know anything about it. Whilst integrating platforms of fundamental sciences, basics, and innovative omics biotechnologies. Just the illustration of omics. Through the algorithms of bioinformatics, including AI, artificial intelligence, which are being implemented finally as the newest entities into the daily medical practice to secure the biodesign-driven clinical, canonical, subclinical, prognostic, predictive, and preventive, newest one, manipulations of the next tech generation. NIH, whilst being a strategic center for American and international medical research and practice, has included ppm associated with translation into a list of the 5 greatest priorities of healthcare services in 21st century. And set up in US 2 academical clinical research networks, including clinical ones, centers of personalized precision medicine. Just a few examples. It's just one of the best in the world, Philadelphia, Perelman Center. Centers of translational medicine to secure the healthcare services armed with technological armamentarium of the next step generation. Few examples. It's the center crucial at NIH. One of the best in the world, the UCSF in San Francisco. And the latest centers of biodesign, one of the best, Stanford. Biodesign Institute in Arizona State. To secure the lifestyle and healthcare services rearmed with the armamentarium of the next step generation. Meanwhile, the worldwide experience and practice say that the efficiency and efficacy of the national bioeconomy are determined and dictated by the innovative trends generated by the freshest knowledge and their transfer into the scientific, bioindustrial, and social area to maintain the national stability and extensive development of the country. So, in daily life and practice, the crucial and indicative sign as litmus tests are becoming through biodesign and bioengineering, or biodesign-inspired bioengineering, biopharma, global biotech, biomanufacturing, and bioindustry. And the design-driven and innovative final bioproducts. It's just the market, illustrating the global market for bioproducts. And innovation is thus considered to become the key to address the unmet clinical needs. In this sense, biodesign, bioengineering, and biomanufacturing can be explained as an approach that includes various research and design-driven applications related to taking inspiration from natural biofunctions, biosystems, biocomponents, or bioprocess in solving the problem. The fundamentals, including synthetic and systems biology, is developing into a biodesign platform where it will be possible to apply the design, build, test, iterate, or deploy to predictably create cells or organisms able to generate a wide variety of novel molecules, materials, or even cells for multiple applications, illustrated here. Accordingly, both of them are commonly used in the design of artificial biomolecules and biostructures in the field of bioengineering, whilst bringing out various bioproducts and biotools that are designed in collaboration with different scientific or engineering and now transdisciplinary disciplines. In this sense, biomaterials are used for the repair, replacement, and stimulation of biosystems. Biomaterials have revolutionized the area of biodesign and bioengineering for the development of new strategies to combat life-threatening diseases and to secure healthcare provision whilst are being used for the treatment of different disorders. Biomaterials are either found in nature or can be created by organisms or can be made in the lab from recombination of starter materials found originally in nature. The recent increased interest in biomaterial innovation is rooted in working within the frame of biosystems through both natural turnover, natural metabolism, and created one. It's just an illustration of the rapid progress of biomanufacturing, which is now becoming one of the drivers of biomaterials trade related trend, including cells, molecules, extracellular or intracellular matrices. To really understand the technological core of PPM, you'd have to understand the various fields of biodesign-driven translational applications that provide the tools to exploit and practice PPM. Not only to exploit, but practice PPM and genomic tools in particular. In this connection, next generation sequencing and NGS is just the illustration of just the mentioned. Hold great promise for unraveling the mysteries of biosystems, gaining broad applicability in disease diagnosis and therapeutics in recent year and future to come. Genomics is considered to be a set of the major and unique molecular tools to probe genome for its quality and now even be tested for predictive and prognostic biomarkers. Just a few examples. Diagnostic testing is used to identify or rule out a specific genetic or chromosomal condition. The results of a diagnostic test can influence a person's choices about healthcare and the management of the disorder. Predictive and pre-symptomatic testing can be helpful to people who have a family member with a genetic disorder or cancer, but who have no features of the disorder themselves at the time of testing. It's just the illustration of some predictive DNA testing systems. Carrier testing, the newest one, is used to identify people who carry one copy of a gene mutation that, when present in two copies, causes a genetic disorder. This type of testing is offered to individuals who have a hidden, latent viral gene pre-integrated into the human genome, or to people in certain ethnic groups with an increased risk of specific genetic condition. Newborn testing to identify genetic disorders that can be treated early in life and other disorders, including monogenic and orphan diseases. Prenatal testing to detect changes in the fetus gene and chromosome before birth to secure the national biosafety. Pharmacogenomics-related testing is aimed at tailoring drug therapy at the dosage that's most appropriate for an individual patient, with the potential benefits of increasing the clinical efficacy and individualized safety. Nutrigenomics is the newest specialty. Last year, only in 6 universities in the world, only in 6. Now, this year, don't know. Is a specialty of the interaction between genes and nutrients. It suggests that an individual's diet should be matched to their genetic makeup, unless and until those tests can be properly validated. They should not be offered as part of genetic testing protocols. The landscape of genetic testing has changed considerably with the emergence of direct-to-consumer DTC genetic testing, like type 1D patients every day. To secure genetic risk prediction tools for a wider array of common diseases and the national health stability. The next important step in the direction of the innovative approach to get healthcare related protection rearmed should be its early adoption in practice for future medical intervention. To achieve this goal, the system needs to rely on data-driven sources of fundamentals, bioanalytical and translational tools and services, and draw from the rapidly evolving high-tech areas and healthcare IT infrastructure. Just the global illustration. Consequently, in coming years, next generation biotechnologies will reorient medical practice more towards disease prediction and prevention approaches, rather than curing them at later stages of their development and progression, even at broader population level for general public healthcare system. It's just the market-related illustration of the value of genetic testing. Value of genomic biomarker market. Due to technologies, due to products, including CAGR. Well, genes can say a lot about an individual's predisposition to a disease, but cannot reveal what's being happened in cells at the proteomic, metabolomic, so-called phenomic levels. Let me stress in the sense that with the advances of biomedical and omics technologies, quantitative proteomics is emerging as a powerful technology complementing genetics. Just the global overview of proteomics. Quantitative proteomics open a wide range of applications in clinical diagnostic, treatment, stratification, and drug discovery. The candidate proteins might provide novel targets for new targeted therapeutic agents or aid the development of assays for disease biomarkers and identification of potential biomarker target ligand drug composites. Schematic structure of receptor targeting drug conjugates. Functional proteomics allows the design of strategies to target multiple pathways with combination of pathway-specific drugs. I will stress, it's the newest one. I guess by the end of the next year, that will be Nobel Prize for pathway-specific drugs, which might increase chances of success and reduce the occurrence of drug resistance. It's just the overview of 3 global segments of proteomics. Meanwhile, chemical from 3 proteomics. It's just an approach by analyzing the drug interactome. It's the newest segment of omics proteomics. The God, when it was born, 20th, 14th, interactomics strongly contributes to accelerate the process of new druggable discovery and drug delivery. By analyzing the drug interactome, strongly contributes to accelerate the process of new druggable targets discovery. It's just the proteomic market size by region, illustrating the value of proteomics. Similar. Engineering manipulation in proteomics, the market. Fortunately, targeted drug delivery can improve the enrichment of drugs at the target size and reduce toxicity to normal tissues and cells. The targeted component is called the ligand. Aptamers, in this sense, have high target affinity and specificity, which are identified as attractive and highly promising ligands. Therefore, aptamers have potential applications in the development of smart targeting systems. It's just the illustration how get it done. Extracellular membrane, receptors, ligands being transformed finally into the drugs, I mean medicines. Meanwhile, aptamers, including DNA, RNA, and peptide ones, are a group of promising recognition units that can specifically bind to target molecule and cells. The specific binding ability of aptamers to cancer-related markers and cancer cells as a whole ensure their high performance for the pre-early, I mean subclinical diagnosis of cancer. Meanwhile, the efficient targeting ability of aptamers to cancer cells and tissue provided a promising way to deliver imaging agents and drugs for cancer imaging and therapy while stimulating the development of molecular imaging. This is the principles of molecular imaging. This is the clinical outcome of molecular imaging. Unfortunately, at this point, only in the area of oncology. Within 2.5 hours, the practitioner would need not longer to confirm 100% cancer diagnosis. No more competing methods. But unfortunately, only few countries have it, maybe 14th or 15th. Much expensive. That's the market illustrating the latter. And finally, companion diagnostics and theranostics. Companion diagnostics and theranostics, its principle of making the therapeutic drug consisting of both diagnostic segment and therapeutic segment. As footstone of stimuli-responsive nanocareers, endogenous or exogenous responsive smart materials possess many properties, such as responding ability to specific triggers, controlled drug release, long blood circulation, increased tumor accumulation. So a combination of genomic and proteomic biomarkers are becoming of greatest significance to be translated into the daily practice to predict risks of the chronification and thus of disabling. Metabolomics. I am not going to unveil the secrecies for you. You're absolutely familiar with that. The applicability of traditional metabolomic engineering strategies has extended with a combination of biodesign-driven systems biology and synthetic biology in a systemic and versatile manner, and are collectively termed, it's an absolutely new trend, systems biology, systems metabolic engineering. At this point, there are only three universities in the world, Yale, Stanford, and Cambridge, who are familiar with that approach. It's just the illustration of the latter. Accordingly, recent advances in biotech have paved the way for enabling the production of an increasing number of monomers and polymers by providing several tools and strategies associated with systems metabolic engineering to develop and manufacture metabolic disorder therapeutics. That's the market, illustrating the latter. Well, but who will patients and now more pre-illness persons at risk and their physicians and biodesigners trust to store and interpret the multi-level data harvested, mined, and used? Healthcare IT do offer a potential solution to surmount those barriers. It's just the illustration of the principal medical information systems, in particular valuable mHealth and electronic medical records, including cloud technologies. Very briefly, modern therapeutic modes and approaches as applicable to PPM. Cellular therapy seems to be very old age, but not yet. I would just concern it later. Gene therapy seems now too very old age, not yet. Just for example, in terms of cell-mediated therapy and gene therapy. 2 years ago, Professor Nick Anderson, not physician, not MD. He is the director of the Center of Cell Engineering of Massachusetts Institute of Technology. He is not talented, he is genius, like Albert Einstein. He has made a population of reprogrammed beta cells capable to generate insulin. He has finalized at this step preclinical in relative clinic, preclinical trial. 108 1,857 chimpanzees with experimental diabetes. All of them were restored in 48 hours. No insulin in 48 hours. You understand me? But 3 companies in the world, greatest companies, Danish, American, and European, have illegally pre-agreed not allow him to start up formally, legally clinical thing. It would be a problem that insulin generating companies. It's understandable. Today, we are experiencing, maybe not all, but most of the research, a renaissance, like in Florence or Rome, primarily driven by biodesign, biotechnologies, and biomanufacturing. And the grand change and challenge to secure our health and wellness are rooted not in medicine and not even in science, in the upgraded high-tech culture. And upon construction of the new educational, training platforms, there would be not a primitive physician created, but a biodesigner or a biopharma or biotech artist to be able to enrich flow-through medical standards with creative elements to give for a patient a genuine hope to survive. For a person at risk, a trust for being no disease. Thank you for your patience. Thank you. Thank you.